Hello everyone, this is part 4 of Unit 3 UGC NET JRF Forensic Science. In this presentation, we will discuss about disputed paternity and maternity, DNA structure, DNA as genetic marker, DNA extraction and profiling techniques. Starting with the basics of DNA structure. So, there is a sugar phosphate backbone on which the nitrogen bases are attached. Nitrogen bases are of two types, purines, adenine and guanine, and pyrimidines, cytosine and thymine. Uracil is present in RNA on the place of thymine. So, in DNA, the nitrogen bases are adenine, guanine, cytosine and thymine. Okay. Now, sugar phosphate backbone. The sugar is a 2-deoxyribose sugar. Nucleoside, there are two important terms, nucleoside and nucleotide. So nucleoside is when the nitrogen bases are linked to the sugar of the sugar phosphate backbone through glycosidic linkage. And nucleotide is when phosphate group is attached to the nucleoside, or the phosphate group is attached to the nitrogen bases as well as the sugar through phosphodiester linkage. Okay, so DNA is a polynucleotide chain twisted in right helix to form double helix structure. Now, Frederick Miescher is the one who identified DNA in 1869, and he named the acidic nucleic substance as nuclein. So earlier DNA was known as nuclein. Watson and Crick gave double helix model of DNA, which we have just discussed. So DNA structure comprises of two chains which runs anti-parallel to each other, both the sugar phosphate backbone. So one is 3' to 5' and the other will go in 5' to 3' direction. Nitrogen bases, there are 4 nitrogen bases: adenine, thymine, cytosine and guanine. Adenine and thymine forms 2 hydrogen bonds with each other, and cytosine and guanine form 3 hydrogen bonds with each other. So adenine form bonds with thymine, cytosine form bond with guanine. There are two nucleotide chains of DNA molecule, and these are coiled in right-handed helix. The pitch of the helix is 3.4 nanometer and each turn contains 10 base pair. So this one turn, this is the complete one turn from this point to this. So it consists of 10 base pairs of nitrogen bases, 10 base pairs of these 4. The human genome contains 6 into 10 to the power 9 DNA molecules per diploid genome. Alec Jeffrey, he introduced a technique known as DNA fingerprinting, in which DNA markers are used for human identification. The blood which is collected for DNA fingerprinting is preferably collected in EDTA, and multi-locus probes are applicable to this technique. Now there is a property of DNA that DNA is somatically stable. So DNA isolated from the skeletal remains gives same pattern as that of the DNA pattern getting from the blood. Maintenance of correct number of chromosome during cell division, mitosis, is through a process known as DNA duplication. There are two types of cell divisions, mitosis and meiosis. In mitosis, from the parent diploid or haploid uh, genome cells, we get From diploid parent we get diploid progeny cells, from haploid we get haploid. In meiosis, from parent diploid cells we get two haploid cells. So now, Oswald Avery's transforming principle. It states that DNA carries heritable traits which transfers from generation to generation. Now, UV rays are known to damage DNA due to formation of thymine dimers. I have already told you that thymine forms bond with adenine. Because of UV radiation, thymine form dimers, it bonds with itself. In October 2007, NIST Standard Reference Material 2372 Human DNA Quantitation Standard was issued. It contains 3 vials of human DNA which is suspended in a clear solution. Mitochondrial DNA. So, human mitochondrial DNA consists of 16,569 base pairs. There is a D loop. There are hypervariable regions HVR1 and HVR2. HVR1 extends from 16,000 to 16,569 base pairs. Cytochrome B gene is used for species identification. There is a D loop that is also known as control region or the non-coding region. The various types of genetic markers are: first is biochemical markers. These markers detect changes in at gene level, such as in amino acids. So, we all know amino acids form proteins and then the chain goes on. These biomarkers are classified as allozymes or isozymes. Allozymes are coded by different alleles at the same locus, whereas isozymes are that perform same enzyme that perform same function but have different structure. So biochemical markers detect variation at gene level. Here comes molecular markers. Molecular markers detect variation at DNA level, such as the variation in nucleotide. So DNA markers are of two types: hybridization molecular based marker and PCR molecular based marker. Genetic markers seen close together on the same chromosome exhibit genetic linkage. Hybridization based molecular markers. These marker examines the difference in the size of DNA fragments. For these markers to work, they require high molecular weight DNA and probe, and these are performed on cellular genome. The technique which I used are DNA or RNA hybridization or restriction fragment length polymorphism or RFLP. In DNA or RNA hybridization, in DNA-RNA hybridization, there is one strand of DNA and the other strand of RNA. The hybrid is used to determine the genetic distance between two species. And in restriction fragment length polymorphism or RFLP, it is used to determine genetic disease, gene mapping for paternity disputes, and is the first technique for forensic DNA analysis. And RFLP analysis determines power of discrimination, variation in the length of a defined fragment of DNA, hundreds of variation at each locus. So, how this technique works? The DNA molecule is cut into many pieces using molecular scissors or restriction enzyme. These fragments are separated according to their length using agarose gel electrophoresis. and then after electrophoresis, the gel is transferred to the nylon membrane via southern blotting procedure. Next, hybridization takes place with radio-labeled DNA probes that determines the length of the fragments which are complementary to the probe. So, uh, in this technique, DNA is cut and then strands are separated, and then using electrophoresis technique, then transferred to nylon membrane, and then next hybridization would take place with radio labeled DNA probes, and we will get the whole DNA sequence. Now, polymerase chain reaction, it is used to make multiple copies from a single DNA strand. So first, the step 1 is denaturing. It is done at 95 degrees Celsius. In this step, the DNA strands are separated. The double helical structure is separated to form single 2 DNA strands. Then, in step 2 or annealing, at 55 degrees Celsius, DNA polymerase the primers get attached on both the strands and new strands are formed with new nucleotides. For example, if we have adenosine here, so thymine would get attached to this adenosine. So a new strand will form, and same for cytosine and guanine. This step is known as synthesis. So we have three steps: denaturing, annealing, and synthesizing. In denaturing, both the strands of DNA get separated from each other. In annealing, primers get attached to the single strands of DNA. In synthesizing, the Taq polymerase will attach to the primer, and the complementary nucleotides will attach to the single strands of the DNA, and two new DNA strands are formed. The cycle is repeated to form more DNA strands. Now, in Sanger method, dideoxynucleoside triphosphates are used for DNA sequencing, as they prevent further DNA synthesis. So, and about restriction enzymes, the natural function of restriction enzyme is to protect the DNA from infecting virus by cleaving it. And these enzymes were discovered by Nathan, Arber, and Smith. Primer extension methods are based on the ability of DNA polymerase to incorporate specific deoxynucleotides. As I have already explained you about the complementary nitrogen bases, and they join together and form a complete new DNA strand. Bovine serum albumins are added to PCR mixture to reduce the effect of PCR inhibitors. They act as binding site for PCR inhibitors. And a stochastic effect or random effect is defined as a variation in fundamental physical law of PCR amplification process. When a very low level of template DNA is there the complications in amplifying results in stochastic effect. Now, what are PCR based markers? So, SNP, single nucleotide polymorphism, in which a DNA sequence variation at single nucleotide is observed. SSR, these are simple sequence repeats or microsatellite or short tandem repeats. These are 2 to 6 base pair of nucleotides are repeated, and D13S317 and TPOX are STR markers. STR markers are preferred for forensic DNA analysis as they are radioactivity based. So in SNP, there is the detection of variation at single nucleotide. In SSR, the 2 to 6 base pair of the nucleotide, it get repeated and used as the marker. In VNTRs or variable number of tandem repeats, the mini satellites or 9 to 100 base pairs are used. In AFLP, amplified fragment length polymorphism, selected fragments are amplified after RFLP technique, and these fragments are visualized under gel matrix to form complete genetic sequence and to establish linkage uh, in case of paternity test or linkage to define crosses, to define the hierarchy. In a rapid amplified polymorphic DNA or RAPD, random sequence of specific markers and random primers are used to develop DNA markers. It is used in genetic mapping, genetic markers linked to a trait in question, population and evolutionary genetic study. Markers linked to a trait in question means whether the trait is present throughout the generations or not. In general, PCR-based genetic systems show less variation per locus than RFLP. Magnesium 2 plus is used in PCR reaction mixture along with SYBR green dye, which is an asymmetrical cyanide dye. How DNA is extracted? So the first is cell and tissue disruption. First step in DNA extraction is cell and tissue disruption, which is done using proteinase K enzyme by boiling or alkali treatment. Bone and teeth are frozen in liquid nitrogen and then crushed. Then the next step is lysis of membranes and organelles. So buffer tris HCl is used to maintain pH. Mercaptoethanol or dithiothreatol is used to inhibit oxidation. Endogenous DNases in presence of divalent metal ion catalyze the hydrolysis of DNA. EDTA is used to capture these metal ions, and sodium dodecyl sulfate and sulfosyl are added for the digestion. Next step is the removal of proteins and cytoplasmic contaminants. The extraction was done using phenol chloroform isomyl alcohol, followed by centrifugation and a reversible binding of DNA to a solid matrix like silica gel. At last, storage of DNA solution tris EDTA buffer at 4 degrees Celsius or minus 2 degrees Celsius for short duration and minus 80 degrees Celsius for long period. So DNA extraction involves 4 steps. First, the cell or tissue is disrupted. Then the membranes and organelles. Then along with the DNA, there are proteins and other cytoplasmic contaminants. These are to be removed. Then lastly, after the extraction of the DNA, its storage. Now extraction of DNA is done using the following methods: phenol chloroform organic extraction method, in which cell and protein digestion is done by proteinase K. Extraction of protein was done using phenol to extract protein, chloroform, isomyl alcohol to reduce foam formation in the ratio 25:24:1. And then after the removal of proteins, the DNA is concentrated by adding certain salts and ethanol. Ethanol and isopropanol are DNA precipitating agents. They precipitate the nucleic acid of DNA. Hence, these are used. Other method is salting out method given by Miller. Salting out of cellular protein by dehydration and precipitation with saturated sodium chloride solution. Then the digestion process takes place. The nucleated cells were taken in. EDTA acts as anticoagulant agent, so it doesn't let the sample to coagulate or to form cluster, and the DNA is extracted overnight at 47 degrees Celsius with SDS or proteinase K. Then saturated NaCl solution is added and centrifuged. This is added to precipitate the protein, and DNA is precipitated by using absolute alcohol, and the extracted DNA is present in the supernatant. Chilex extraction method, which constitutes of 3 steps: washing to remove contaminants and heme, boiling with 5% of Chilex 100 chelating resin, and centrifugation at pH 10 to 11, temperature 100 degrees Celsius, and this supernatant will contain single-stranded DNA. Silica-based method, cell lysis and protein digestion was done using proteinase K. DNA absorption into silica. Binding will be in the presence of cheotropic agents. Washing of DNA was done using ethanol-based wash solution to remove the cheotropic agents and other contaminants. Elution of double-stranded DNA was done by using low aqueous salt solution. Then differential extraction is done usually in rape cases where there are male and female, both the cells are present, male sperm cell and female epithelial cells. So based on difference in mechanical strength of the cell membrane, as the sperm plasma membrane contain proteins cross-linked by disulfide bonds. So the male sperm cells will have high mechanical strength. Based on the difference in mechanical strength, there is differential extraction. Non-sperm cells are lysed in the presence of SDS and proteinase K, and the removal of non-sperm cell is done using centrifugation. After separation of non-sperm cells and after extraction of DNA, it is amplified using PCR. Visualization on agarose gel is done by electrophoresis. Dyes that intercalate with the DNA double helix, such as pica green or ethidium bromide, are added to the gel before or after electrophoresis, and DNA is visible under the UV light at 260 nanometer. Slot blot technique, which involves visual comparison of two DNA strands based on the band intensities. It is based on specific hybridization of a 40 base pair probe which binds to human alpha satellite locus D17Z1. Real-time PCR is proposed by Higuchi by including ethidium bromide in PCR reaction so that the DNA is visible in the UV light and it can be monitored continuously. DNA IQTM, it is based on salting out and binding to silica, both the methods. A very specific amount of silica coated beads are added to the extraction to bind a maximum amount of DNA. First, DNA is extracted from using salting out method, and then silica coated beads are added. The silica coated beads will increase the amount of DNA the silica coated beads would bind the DNA and increase its amount. Therefore, when the DNA is eluted, maximum concentration is known. DNA multiplexing, analysis of different loci of DNA simultaneously. Stutter band in DNA analysis, it is the consistent observation of minor band. So it is 1 unit smaller and it is present after a real peak. There is small peaks. So these are the stutter bands. In SDS page or sodium dodecyl sulfate polyacrylamide gel electrophoresis, the ionic detergent SDS is used to feature and bind the protein to make them uniformly negative charge. We have already discussed that protein digestion is done using SDS and proteinase K. So SDS, what it does is it binds the protein and make them negative charge, so that in SDS page proteins are separated. The factors responsible for migration rate of DNA in agarose gel are the size of DNA. The smaller the size of DNA, the faster it will travel. Conformation of the DNA, ionic strength of the running buffer. Size, charge, conformation, ionic strength. Now the various types of blotting techniques. Southern blotting, it is the procedure for identifying specific sequence of DNA. Northern blotting for RNA. Western blotting for proteins. And Eastern blotting for post-translational protein modifications. Disputed paternity and maternity is identified using DNA markers. We have already discussed about it. DNA profiling, application of ABO grouping in disputed paternity was first observed by Ottenberg. Human leukocyte antigen typing or HLA typing is developed by Dr. Paul Teraski. He discovered this system to minimize the possibility of rejection of transplanted organ. Leukocyte is what? WBCs. So during transplantation, the rejection should be minimum. Because of this particular thing, he have developed HLA typing. So for HLA typing, it is a protein test for HLA-B27 proteins. HLA are highly polymorphic glycoproteins on the present on the surface of WBCs. Mendelian law. What is Mendelian law? Mendelian law states that individuals who possess two alleles that get inherited by two alleles, one from each parent. Blood group and HLA in offsprings are determined by Mendelian law. Like Mendelian law, what it states that alleles, the two alleles, one will be from the mother and the other will be from the father. So blood group, it is also made from 50% from the female and 50% from the male, and HLA is also the same. So in disputed paternity and maternity cases, this can be used. Paternity index, it is calculated value generated for a single genetic marker and is associated with the statistical strength or weight of that locus in favor of or against percentage and inheritance scenario. Paternity index. Based on these tests, there is a calculated value which tells us whether the particular person is the parent of that particular child or not. The various DNA locus and the chromosome number on which these are present are FGA, human fibrinogen alpha, is present on chromosome number 4. THO1, human tyrosine hydroxylase, is present on chromosome number 11. TPOX, human thyroid peroxidase, is present on chromosome number 2. VWA, von Willebrand factor, is present on 12. CSF1PO, CFMS proto-oncogene for CSF1 receptor gene is present on chromosome number 5. The various techniques and the year of their discovery are HLA-DQ 1990, first STR kit 1993, D1S80 or amylogenin 1995, mitochondrial DNA 2002. HLA-DQ is human leukocyte antigen, first STR short tandem repeat. Amylogenin is used for the identification of whether it's a male or a female. So there is 6 base pair difference between these two. We'll discuss about it in unit number 10. mtDNA is mitochondrial DNA. Now, Wildlife Protection Act is given in 1972. There are few amendments of WPA. In WPA 2002, the amendments were related to punishment. WPA 2006 related to saving tigers and other endangered species. WPA 2013 punishment for hunting and hunting rights of scheduled tribes in the Andaman and Nicobar Islands. Thank you for watching. Stay tuned.